So I made a little presentation to start with. Should I hide this toolbar? Can they also see it?
Yes, we can see the toolbar, but okay, I think it's okay.
Then hide. There.
Okay.
Okay, so we will start with registry. So there are 5 stages that you'll find yourself loving or hating the registry. I personally hate it. So the first stage is the what. You have no idea what it is, probably. I also didn't. The second stage is when you go to the website and you find out that this website is really not just 2006, probably 1990. It's very, very bad. Stage 3 is how the hell does it work? And actually, that's the stage I hated the most because when you start adding parts to the registry or trying to do any type of work with the registry, and nobody has told you how things work or what you should do, you become frustrated. And you become extremely frustrated when there's only 4 days left to submit your parts. And my recommendation is to not wait until the last day to submit the parts to the registry. That's what we did. And it wasn't fun at all. And the stage 5 is that you cannot even remember what you submitted. While I was making this presentation, I was trying to remember what we submitted to the registry, and I have no idea what happened. And I think it's probably because of the panic mode of the 4 days left to submit your parts. Anyway, hopefully you won't go through this entire cycle, but most likely you will. So, why we need the registry? I guess for all of you, it's quite obvious that we need the registry to exchange parts, to document how they work or what we can do with them. And of course, you can access it through this website. We will go there in a bit. So there are good and bad examples of things in the registry. So we'll look at the good example from a team. This is not working. This should work. Yay. I'm a little fanboy. Actually, most people from iGEM Athens 2018 are a bit fanboys of Heidelberg teams. Anyway, so this is, in my opinion, this is a nicely documented part, although they could have done more experiments to document it, but you can see everything is quite well explained, the usage in biology, what it is. They made some nice graphics, how things work. And they also did some measurements, so they also have both qualitative and quantitative data. Yes, so what I don't like is maybe the fact that they haven't annotated the sequence down here, but more or less this is a nice example of what you should do. There are also extreme examples of like super good documentation, but I think in the time frame of iGEM, this is a very nice example. And also from last year, uh, the the guys in Thessaly, they did nice documentation of their parts. So I also added it here, support our Greek teams in a way. So you can see they also added a nice explanation and a nice quantitative experiment. And they also added the wells. So you can actually see the color changes in case you're doing the experiment yourself. And also down here, you can see the different parts. That's what I meant by annotating the sequence before. So in this case, you can see the parts that this sequence is consists of. Okay. And the bad example, I picked myself. Um, okay, so it's a nice description, let's say, of what this thing is, but there's no experiment, there's no actual data. But in many cases, many people submit parts without any experiments. But it would be nice, as in this case, for example, to add a good description and the references uh, to support what you're talking about. In this case, this is an enzyme that we took kind of from a published paper. So we cite it here in case someone wants to look at the paper itself, because this is a structural paper, and the protein structure was what guided us to mutate it. Okay, so before you submit a part, so before we go to look at the registry in more detail, there's two things that is important to do. First, search if the part has already been submitted, and I cannot stress this enough. It's a little bit annoying. Right now, for example, we were making some catalogs for aptamer sequences, ribozymes, aptazymes, and stuff like that. It was a little bit annoying that a part existed and then teams kept on submitting the same part for, I don't know, 5 years in a row. This should not be, this is not acceptable actually by it shouldn't be acceptable by the regulations of iGEM, let's say. And I think the registry, they started to discard duplicate parts, but it's very easy to just search for the part. Maybe someone has already submitted it, because if they have submitted it, you can use it to, I don't know, provide more characterization, um, or I don't know, use it in your system without having to do the characterization yourself, and all these kind of things. And the second one is that if you decide to submit a part, you should understand what it is. So that's, I have like some questions. So just think about if it's a basic or a composite part, and we'll talk about them in a minute. If it goes for DNA, RNA, or protein, if it's a functional nucleic acid, and if it's a protein, what is the function? The last two are quite important, and I will tell you why when we look at how we add the part. The third one is not there yet. We're working on adding a category of functional nucleic acids, but in case you're interested, keep it in mind. So many of you probably already know what is a basic or a composite part. And also in the registry, they stated what is a basic and what is a composite part. So here's my like 2 bullet points. If the part encodes for one single sequence, it's basic. If it encodes for multiple sequences, it's a composite part. So you see here, for example, that we have part A, that is lysozyme, which is just a protein sequence. Then it's a basic part. And then you have a part B, which has the RBS only. Then this is a basic part. But if you have another part B that has a promoter plus your lysozyme, then this is a composite part. Maybe you think this is a bit stupid, but actually when you start looking at the registry, a lot of people don't submit parts correctly. So now we'll look at a live demo. These were supposed to move, but they're not moving. If you have any questions right now, you can ask. Because I'm speaking to my screen and I don't like it. Anyway, so this is the registry. So if you go to parts.igem.org, you don't have to type the /main page. It redirects you. to this page. This is the registry. And that's what I mean by this is 2006. It looks not functional, but I'm not here to judge the registry. Okay, so you will see you have a toolbar up here. Also here above, it's super, super, super small. But before you do like things in the registry, it's nice if you can log in. So now I logged in. Okay. Okay. So you see here we have a few menus. We call it a menu. And you see we have a catalog. So one of the pages I like is the very well documented parts. So you see here they made some tables, and actually you can click on some of them and you see pictures, characterization, blah blah blah, all these different things. Please keep in mind when you look at these pages that it's not just one team that has done all these experiments. Up here, you see who has submitted the part and who designed it. But you see most of the experiments come from other teams. Like this team last year provided the characterization for this part doing this experiment, and so on and so on. But it's nice to come to this page when you want to design your experiment, for example, to characterize your part, to maybe, I don't know, be creative or find some inspiration for how to design your experiment. Okay. Then you can also browse by type. So for example, you can look at promoters and you have there is a promoter catalog. You can look at constitutive promoters. Yes. So you see here you have the part numbers, you also have the sequence, you have some information about different parts. Okay. And in the catalog, there's also the different collections. So for example, maybe you want to use CRISPR or CRISPR-Cas9 systems. It doesn't have to be just Cas9. Maybe you want to use, I don't know, Cas12a or whatever. So here they have collected, not here, here they have collected the different CRISPR type of enzymes, which is, in most cases, it's complicated, so it's probably not good to use. But you also have some others that are probably a little bit more well characterized. So I'm showing you the catalog because maybe from one of those collections you want to use some parts in your system. And the other one that is important, and I'm looking for it now, it's the distribution. So this year, the teams will not receive a distribution kit. They will receive some parts that they have asked, or I think part of the distribution kit from last year, something like that. Hopefully next year, you or I don't know, someone you know will receive the distribution kit. And then you want to know what is where, because the distribution kit contains, I can't even remember, what is it? 2,000 parts, 3,000 parts, something like that. So you want to know what's in each. Well, so you see, you can just click on the plate, and you can get the antibiotic files. It says here. So you see for each well the the part that is in the well. So in well 1A of plate 1 from the distribution kit in 2019, you have the F1 origin. And you can also look at the distribution kits from other years. For example, in Athens, we still have the distribution kit from 2018. So if people want to use it, they can go to the website here and find out what is where. Usually, most of the parts are the same. So you see here, for example, it's again the F1 origin, but some parts are different between the distributions. Okay, and the registry also gives you some information about assembly. You have the different standards, so you can read about, for example, RFC 10. There's enough information. And you also have some protocols. So maybe you want to do the classical 3A assembly. They provide you with a protocol here. Okay. So if there are no questions up to this point, probably not. So in order to add a part to the registry, you go to Tools, General, and Add a part. So you click Add a part. In this page, and also in the other pages, maybe you noticed, you can see the help pages up here. So at any time, you can click at the frequently asked questions, for example, and see maybe you have a question regarding, for example, adding parts, because that's what we're doing right now. So you can click adding parts, and it will tell you some, they're useful. The questions it has, the questions and answers they have here are useful. Okay, so you can see here we have the option between basic parts and composite parts. And if we want to start with a basic part, you see that you have to add some information. So you need a part name, a description, well, actually a short description and a full description, the source of the part, and that's it. So in your case, it will tell you the team that you belong to and the part range that you can use. So because I made a part with the last available name, now it tells me is already used. But you can see up here, because I haven't added a part yet with this account, you can see that it already tells you what is the next available part. So this is the code that you can use for this part, and you can add it here. In my opinion, it's good to click to tick the box here to allow edits, so people can go to your page and edit it in case they find something wrong or they want to add characterization or I don't know what. Here you can add the description, so we can already add a part. I have added one, but let's add another one. Also here you see there is a part type, and this is important. So right now we won't add something interesting, but you can see that you can already specify that this is an RBS, this is coding for something, this is a reporter, this is a terminator. For us, this is temporary. And you can just put the text temporary part example. Usually when you're in a rush, you won't add anything here and here. So you can add no, no, no to continue to the next page. but it's best if you already add some sort of description here. So you can see uh, in the long description, you describe like the usage of this part, where it comes from, what it does, etc. Um, Here you have to add the source of this part. So maybe the gene or the protein you're adding here comes from plants. So it's good to say, for example, that this is coming, I don't know, from olive trees, and add a reference from a paper, maybe you found it. And some people add design considerations. So for example, maybe you found an early stop codon for the organism that you want to express this. So you had to change, for example, I don't know, this nucleotide at this position, or you wanted to link to different domains. So you designed a linker, and you can add this design consideration here. For the most parts you see on the registry, and really just go to the registry and look for parts, people do yeah, poor submission of parts, so you won't see them adding stuff here. So we go to the next page, and you can see that now we can add, it tells us that this is the part that we have, and we can add the sequence. You can just click edit. So I'll just uh, take a sequence from here and just add it here, and I can save. So now I have my sequence here. Here you can see also the sequence. You can also do the double stranded, but it's not very useful. And it can already tell you if the part that you're submitting, the sequence that you submitted, is compatible with, I don't know, which assembly you want to do. Maybe you want to do RFC 10, maybe you want to do RFC 1000. So it will already tell you, for example, if we had an eCore 1 site, maybe we can go introduce it somewhere. You see, it automatically found out that we have the eCore 1 site, and it tells us, oh no. So for example, maybe your part contains this site, and in your design considerations, you can say that there was an eCore 1 site. So I had to change the nucleotides. You can make a missense mutation. Yes, no, yes, probably. I'm not sure. Please forget what I said. It's not a missense mutation. I'm not sure. In any case, you can just exchange for the same amino acid. So you can find the genetic code, and yeah, in the end, the protein won't change much. Or anyway, okay. And here you can add features. So if we say add a feature, maybe we want to say that it has an RBS, and we can label it as RBS. Okay. From 1 to 100. And it tells you also in which strand, and you can click submit. So you see now it has annotated down here what this part of the sequence is. In my opinion, it's cool if you do that, because when someone goes to check your part, they can have an idea about uh, the sequence and what they're looking at. So we save. And then there's many things that you can do. So if we look at the main page, you see the title here. It's a temporary part example. We have the part number here, who has designed it, so who submitted it and from which team and when. And when we go to the design, You see there's no design notes, but I can edit and add. If we go to the experience page, there's nothing. So this is where usually you will add the characterization of your part. And if it worked as expected, stuff like that. And in the information page, you can find some more information about this part, but you can also add some parameters. So for example, maybe you want to say this is, let's see what it is, an MCS. So it has parameters as an MCS, and you can also have a category. So remember the catalog I was showing you before with the different collections? That's where they come from. So if we can find Cas9 here, you can see they have many different things. So we won't find it, but they have it somewhere here. In any case, it's good to do this because the tables are automatically updated. And for many things, maybe, for example, you want to ask the registry to make a part collection with, I don't know, the different, I cannot pronounce this correctly ever, sassis. I always call it in a different way that in Greek means something very different. But yes, if you check this, then the tables that contain this kind of stuff will be updated to contain this part too. Okay, and in the part tools, you can find some more information. You can find related parts in the registry. So you can see there are some related parts in this category. It tells you that this part has no twins, which means that nobody else has submitted the exact same sequence in the registry. Um, and at any point, you can come to edit the sequence and all these different things from the part tools menu. If you go to the edit, you can edit the main page. So here, for example, in the usage and biology, you can say that this is cool part, and blah blah blah, and you can save. Awesome. Usually it saves stuff. Now I don't know why, but anyway. So that's how you submit a part. It seems easy, but the difficult things is when you come here and you have to write a lot of different things, and maybe you want to add images. So the way to add images to the registry, it's the exact same way you add images to your wiki. And I don't know if someone is going to talk about that, but I don't fully remember how to guide you to do that right now. It's not difficult, it only takes a little bit of time, and don't do it in the last few days. Okay. And if we go back to add a part, and we can go to the composite part, we can submit a composite part in the same way that we had before. But here now you can add the names of the parts that make up this part. So we can already say that I want to mix this Oh, this is the other part I was making. With this, maybe. Please ignore the stupid titles. And you can generate this part with no scars, depending on the assembly you're doing. I did something wrong. Okay, I'm getting an eternal server. It won't, I don't know why it's not, it doesn't want to collaborate with me. Okay, I'm really sorry. I don't, it's, what is the problem? I'm sad. In any case, you can add the composite part in the same way as you do with your basic parts. In the sub parts, I don't know, I thought it's a colon. Anyway, you can add your sub parts here. Because then it will automatically annotate them. Yeah. It won't let me move on. But if you add the subparts, then it will redirect you to the pages that we saw before. So you can add more things. You can add descriptions, characterization, the category it belongs to, and all these different things. Okay. Um, if you have a question, you can ask because I'm gonna go back to my presentation to finish. So I will finish. So to recap, you should understand your part. So as we said before, what it is. So is it a basic or a composite part? This is important because there are different buttons to submit those to. You should know if it codes for DNA, RNA, or protein, and what's the function of the thing that it is coding for. Please write down everything you know about this part because you're the person that is using this part and probably knows the most about it. And 2 years from now, or even 6 months from now, you won't even remember what this thing is doing. And I'm speaking from experience. Don't wait until the last day to submit your parts. And this is important not because you're stressing out, but as you also know with the wiki, the system is failing. So at some point, it takes you probably half an hour just to go from one page to the next. And you can always edit the part later. So you can already submit your parts, write down a few things, and go back to edit those pages. So I will leave you with this slide of ours. That's me, Fanny, and Jason. This is the last day of the wiki freeze. I'm drunk, sleepless, and we finished at 7 a.m., which was the last, it was the deadline for the wiki freeze. I was submitting parts on that day, and Fanny was making some edits to the wiki. So that's it. I hope it was a bit useful. Please ask questions. And how do I go back?
Awesome. Thank you. Yes, does anyone have any questions?
It lasted more than I thought it would.
No,
I have a question. So for this here, do teams have to just edit or add onto existing parts?
That I don't know, because usually the registry is part of the judging criteria. So I really don't know.
I don't know as well. That's why I'm asking, in case anybody here knows.
I guess we'll find out next Thursday. Anyone else? Would you recommend being drunk for the wiki freeze?
No.
No.
I mean, it was fun. It was definitely fun because